WorldCommunityGrid.org has been harvesting spare processing time from computers around the world for nearly 7 years now, using that computing power to accelerate research to help humanity and the world. We owe great thanks to our members for contributing this time to the important projects we have been helping. One of the research projects on World Community Grid is the Discovering Dengue Drugs Together project from the University of Texas Medical Branch in Galveston, Texas. They have been running their research on World Community Grid since August of 2007 and are in the second phase of their project to search for potential drug candidates that could fight a class of diseases including dengue fever, West Nile virus, yellow fever, and hepatitis C. Please let me introduce Dr. Robert Malmstrom, who received his PhD through work on this project. He will tell you about the science behind the Discovering Dengue Drugs Together project running on worldcommunitygrid.org.
Thank you, Victor. This is Robert Malmstrom, and I'm going to be discussing our project, Discovering Dengue Drugs Together with you guys today. I'd like to start off by thanking all the members of World Community Grid, because without you all, it would be impossible to do the work that we're doing. So to get started, I'd like to talk about dengue itself and give you guys a little bit of background in it. Dengue is a mosquito-borne virus. It causes dengue fever in a lot of people. It can also progress to a hemorrhagic fever, though not quite as violent as a bullet, still a very, very serious disease. And if that progresses poorly, it goes into what's called dengue shock syndrome, which is fatal. Dengue threatens 2.5 billion people annually, and that number continues to grow as the mosquito populations spread farther and farther north. There are about 50 million infections annually. 500,000 of those people develop the hemorrhagic fever, and there's 22,000 deaths annually. And most of those are from susceptible populations like children. So we've been very interested in trying to develop treatments for dengue, because currently there's no vaccine and there are no antiviral treatments. And so we've been our lab is real interested in finding antiviral drugs to combat dengue infections in humans. So drug discovery is not an easy process. It's very time-consuming. It can take up to 15 years to take a hit all the way through to a new drug challenge. And we sit at the very beginning of this process, and we're interested in finding hits and finding compounds that can be used as treatments against dengue. And we have selected the virally encoded protease. The dengue virus encodes its own protease enzyme, and that if we knock that out, we inhibit it, it cannot replicate anymore. And so we're looking for drugs that can inhibit that enzyme. And so when the drug discovery process starts by finding hits, and that's what we do. And those hits are then fed into the drug discovery pipeline, which is quite a challenging process. It's only a little less than 1% of the drugs actually get from discovery through development into clinical trials. And even in clinical trials, there's only a 20% success rate. So we want to be able to find as many potential compounds and explore as much of chemical space as possible. So there are multiple methods for finding hits. There's high-throughput screening, where you take robots and run assays in mass through very large chemical libraries. There's virtual screening, where you do a similar process but computationally, and that's what we're doing. There's scaffold hopping, where you take known drugs and you modify them. And rational drug design, where you use the actual target, in our case would be the protease, and you hand build new compounds and test them, or it's through natural products discoveries. So we're really interested in the virtual screening process, because virtual screening allows you to test a greater variety and explore more of chemical space than you could with a high throughput screen, and lets you do that before you even have to go to the bench. Now, how does virtual screening work? Well, the workhorse of virtual screening are what's called docking programs. And docking programs find the geometrical relationship between a compound and our biological target. And so here's an example of possible outputs from a docking program. And docking programs are composed of basically two functions. One is a search function that explores the conformational space, or trying to figure out different ways that the compound can interact with the target, and a scoring function And the scoring function evaluates the fitness of each of those poses. And as you can see here, I've have a bunch of poses put up, and I've color-coded them according to the fitness, with the green being the best, to kind of give you guys an idea of what goes on in a docking program. Now, docking programs are very good at reproducing experimentally determined poses. However, their scoring functions are inaccurate in predicting the actual free energy of binding. So though they can kind of figure out what the pose is, the actual total energetics of binding, which is more complicated than just the geometric relationship between the ligand or the compound and its target, is not well predicted. But even with that inaccuracy, docking programs are still effective tools for what we like to call enriching chemical databases. And so here is what's called an enrichment plot, or we call a success plot, for a retrospective virtual screening project that we carried out. You can see that on the x-axis we have the number of compounds in the database, and what that is the compounds in the chemical database rank ordered based on the score that we get from a docking program for this particular case. And you'll notice also it's a log scale. Just to remind you that on the y-axis we have the number of known binders in that data set, because this is a retrospective study, and you can see that ideally, with 70 binders, we should find all 70 binders on the top of the list, and we don't get that with most virtual screening projects. It's less than that. But we usually do better than random, so we can truly enrich the database. In this case, if we looked at our top 100 compounds, if we were to take those to the laboratory and test the top 100 compounds, we would get about 10 hits. And as you know, we need lots and lots of hits. So 10 is a good start, but we need to definitely improve on what the enrichment that these docking scoring functions are giving us. So what we've proposed and what we do in our Discovering Dengue Drugs Together project is a two-phase approach. And these phases correspond with the two phases that we had in our Discovering Dengue Drugs Together project on Rural Community Grid. So phase one, which we've is pretty much completed, was a standard virtual screening project with DOC. And so we had a database of 2.3 million compounds, and we docked those compounds against a variety of targets, and particularly our dengue targets. And so what we're going to do is take those poses, and then we're going to apply them to a more rigorous method for determining the free energy of binding. And that's what the phase two project has been about. And I want to talk a little about the results of phase two today and kind of the exciting science that's come from that. So we have these, so we're really taking the poses and stuff from phase one and processing them in phase two. So how does the phase two calculations work? The free energy of binding calculations are, as I said, computationally very, very rigorous. And in kind of a quick method, a quick way of describing them, what we use is molecular simulations that allow us to explore the energy landscape around the docking pose, and also to explore the energy of the ligand in solvent. So what we can determine from doing all these little molecular simulations, which is what we actually run on the individual computers in phase two, are these individual molecular dynamic simulations, we can look at the energies as we slowly perturb the system or remove the ligand from the site, and then we can determine what the energy of binding to the site is. And we also do the same thing for the solvent, because binding, of course, is, you have your ligand and solvent, your protein and solvent, and you bring the two together, and that's total energy of binding. So we can use all these little simulations to determine the free energy of binding. And these calculations are, say, computationally rigorous, but generally prove to be more accurate. So why we ended up using the grid for what can be good for this is because, well, it is computationally demanding. If we compare the docking calculation to a free energy of binding calculation, for one ligand it takes about an hour to run. We generally did about 5 compounds per work group with you that we would run on your computer. Now, for the phase two, just to get one free energy of binding calculation done for one ligand, it takes 21 days serially, and that's on kind of an older processor I had when I did the timing. but it is very, very computationally demanding. But fortunately, this isn't a serial process. We can break it up, and we can use the web to run the different parts of it, because each MD simulation that we run can be run independently of the rest, and that's what we run on your computer. And that allows us to do very considerably larger free energy binding calculations than ever before. Because of the computational demand of these perturbation stages, most previous studies, including our early studies in this work, would be at most 60 compounds. That would be a really large database. And now we can actually process things in groups of 2,000. So here is a retrospective study that we did early on to test and really make sure that everything was working. And you can see here that when we do the free energy binding rescoring from the docking phase, we get an improved enrichment over what we have for the free energy scores from what we have from docking. And so in a real practical sense, if you think about what this means in the lab, is if we sit down and we take the top 100 compounds from our virtual screening projects, right? From the docking, we would only get 10 active compounds. Well, if we do the free energy binding rescoring, we end up getting 30 compounds, get about a threefold increase in the number of possible binders that we have. Now, as we do our work, of course, we're doing this in a prospective way, and so it really is an issue of kind of hedging your bets and trying to get the best results possible. So as we apply these free energy calculations to rescoring the stuff we do in from the stuff from phase 1, then we're filtering our database and increasing the probability that we'll be able to actually to efficiently find drugs or compounds or hits that we can develop into drugs. So where are we now? So with discovering dengue drugs together, our targets being dengue and West Nile, hepatitis C, and protease inhibitors, we've completed phase 1, which consisted of 16 different virtual screening projects. And thanks to you all, we were able to rack up 11,000 years of CPU time in that. Phase 2 is currently active, and hopefully some of you have work units now and are processing those. We've been able to do about 4 rescoring sets. Each of those are around 2,000 compounds. So if I remembered earlier, I told you that we were only able to do 60 compounds at a given time, 60 free energy scorings. Now we can do 2,000 in just a few months. And that project, of course, is accruing CPU time very rapidly. Additionally, we're doing a related project, which is also on World Community Grid, the Influenza Antiviral Drug Search Project, which is looking at potential neuraminidase inhibitors. And that project's on hold, and we'll advance that one to phase 2 as we complete our work with discovering dengue drugs together, phase 2. So I thank you all for your time, and I really thank the members of World Community Grid for providing their computers, because without them, we would not be able to do these calculations. We've been able to truly do these calculations in a scale that is unprecedented, and really analyze and see how well we are doing in our ability to predict the free energy of binding. And the results are encouraging, and also give us, of course, as above science, gives us way more questions than answers. I'd also like to thank Benoit Rowe and Yuxing Ding from University of Chicago. They're the people who originally developed the free energy method that we're using, and have been good collaborators for us. I need to recognize the Texas Advanced Computing Center. It takes a supercomputer to even prepare the work for you all, and they're the ones who provided a lot of that computational power for us. The Wadowich Lab and our drug discovery group. All the people, of course, at IBM, World Community Grid, who've made this project possible and let us do basically something that was impossible before. And, of course, all the different funding organizations that we've received funds from over the course of this project. Particularly the Golf Course Consortium, which helped funded me with a training grant as I finished my doctoral work, and the IBM International Foundation for helping us do our compound testing. So thank you, and I turn the time back over to Victor's.
Thank you, Robert, for a very enlightening talk about the Discovering Dengue Drugs Together project running on World Community Grid, and increasing the awareness of how we are all helping with the spare time from our computers. To those listening, thank you for attending, and I hope you will all contribute your spare computing power to worldcommunitygrid.org if you have not already done so.